Hello and welcome to this brief introduction to the first clue for drug repurposing. When we want to develop a new drug from scratch, we need around 10 to 15 years of research for preclinical and clinical investigation just to make sure that the drug is safe and has efficacy. On top of that, the cost is also very huge. Bringing a new drug to market can cost around $2.5 billion when we account for failed trials, attrition, and regulatory requirements. But the interesting part is that we already have thousands of drugs that are approved for a specific disease, and many of these drugs can show pleiotropic effects, meaning that they can modulate multiple biological pathways beyond the pathway or target that are associated with their original indication. Sometimes these secondary effects can cause unwanted side effects or adverse drug reactions, but they can also point to new therapeutic indication that can be completely different from the original indication of the drug. And this is what is called drug repurposing, that we find new uses for drugs that are already approved, which is of course a faster, more cost-effective, and generally lower risk solution compared to de novo drug discovery, because these drugs already have well-characterized pharmacokinetics, pharmacodynamics, and safety profiles. So if we do repurposing, a big part of the development pipeline is already done. A classic example for this is imatinib. It was originally developed to treat chronic myeloid leukemia by targeting the BCR-ABL tyrosine kinase, but later it was repurposed for gastrointestinal stromal tumors. Okay, now the question is, how can we systematically find these new use cases or do drug repurposing? Historically, many repurposing discoveries happen by chance through clinical observation, off-label use, or unexpected effect that's seen after the drug actually were on the market. Also, we had important clinical advances with these serendipities. Relying on chance observation is not efficient, especially because most of the time they are biased toward well-established diseases where there is more data and larger patient populations. So there is a clear need for systematic and scalable approaches. Okay, this is where computational drug repurposing comes in. With these methods, we can integrate large and diverse biological data sets to prioritize potential drug disease associations and also generate testable hypotheses for the clinic. There are various computational methods for drug repurposing that rely on different type of prior knowledge and different assumptions. But in all of these methods, we always start with genes or proteins that are already suspected to be associated with a particular disease. These genes are called seed genes. They are our starting point or our first clue for understanding the mechanism behind diseases. So finding relevant seed genes is a very critical step, and there are generally two ways to do this. The first method is database driven, in which we use existing biological knowledge from curated databases. For example, databases such as DisGenet or OMIM that provide disease gene associations for different type of monogenic and polygenic diseases. But the issue here is that for rare or neglected diseases, there may be very little information available in these resources because not all of these diseases are well studied. And the second method is data-driven, that with advances in high-throughput technologies, now it's possible to measure gene expression, proteomics, and other molecular features at larger scale. So instead of relying on existing knowledge, we can generate and analyze a new biological data, for example, from patient samples. And in order to obtain seed genes with this data, for example, we can compare tissue samples from disease and healthy individuals, and then by doing so, we can identify genes that are significant significantly upregulated or downregulated, that these genes are called differentially expressed genes or DEGs, and they can be considered as our seed genes. Once we have these seed genes, we can move to the next step. Typically, classical network-based drug repurposing methods follow two main steps. First, we identify disease-associated modules in the interactome. We start with seed genes and then expand them through protein-protein interaction networks to find groups of genes that are functionally related and represent the underlying disease mechanism. And second, where we prioritize drugs based on network proximity or other criteria. The idea here is that if a drug targets proteins that are very close to the disease module in the network, it is more likely to influence the disease and potentially have a therapeutic effect. The next videos go deeper into some of the most commonly used network-based computational approaches for these two steps for identifying disease modules and also drug prioritization. Thank you very much.